I mean, I think it's very significant. Right now, interstitial lung diseases, pulmonary fibrosis, when we identify these patients and start treating them, they already have very advanced disease. And so these are people who have a lifespan that can be counted in years, single digit years, 3 to 5 years, something like that, by the time we actually find them. So the, it's significant in that we're trying to identify these people and intervene so much more early that they are able to hopefully avoid some of the long-term consequences of developing pulmonary fibrosis. Yeah, I think that's the answer is that there are no approved options for these individuals. And quite honestly, we're not entirely certain what to do with them, right? When we see them, we tell them, you might have something there and it might turn into a disease later on, but we can't give them much more than that. And so this is one of the first studies to let us start to understand what that aspect of the disease, the very, very, very early disease, how that behaves and whether intervention with a known effective medication will improve their outcomes. Yeah, we think it's pretty important for a couple of reasons. The first is that most physicians believe that imaging in the form of HRCT for ILDs, imaging changes almost always precede functional changes, the breathing test, the FVC that we use. And so we believe that if we really want to detect the very earliest signs that things are getting worse, we need to use HRCT. Um, the the nice thing about using a quantitative HRCT is that the computers can see changes in the images well before the human eye can. And so we think it's important. We also think it's important because as more and more drugs come to market for treating patients with pulmonary fibrosis, um, our uh, our ability to impact the change over time in breathing tests and forced vital capacity is diminishing. So it's going to be harder and harder to determine whether a drug is doing something for a patient purely based on the forced vital capacity. So in terms of biomarkers, really the only biomarkers we're using are the imaging ones, is the CT scan. So within the e-lung software, there is uh, certain metrics. There are certain metrics, weighted reticulovascular score, total disease extent, and so on. So we're going to be looking at some of those. We will be doing a little bit of blood biomarker work, but purely as an exploratory study. So nothing that would impact the primary outcome. Right. The the physiology of the lung is that forced vital capacity measures the amount of air that can come out of the lung in the first or overall. So you take your deepest breath in and you blow out as much air as you can until there's absolutely nothing left for you to get rid of, and that's your forced vital capacity. The forced vital capacity is impacted by how much air can fit into the lungs. So the imaging study will show us what parts of the lungs are being destroyed before it impacts the amount of volume that comes out. And so because of that, the CT scan, the imaging, should give us quite a bit of information before we even get to the point where people are starting to lose lung volumes. We're pretty confident that the AI assisted CT quantification will have very significant clinical meaning. There's already some data demonstrating that changes in some of these quantitative biomarkers over time predict uh, worsening of lung function, predict mortality, predict uh, um, a poorer outcome overall. So I do think it's going to be pretty clinically meaningful. When it comes to brainomics and their e-lung technology, excuse me, Boehringer has had a fairly long relationship with them. We've done a bunch of work. We have a few abstracts and a paper coming out shortly looking at some of our prior clinical trial data using their technology. And it is pretty reproducible. It's very robust. And I can't stand here and say that it is the best one out there, but it certainly seems to be very consistent and it seems to be very easy to understand. And it also has FDA clearances in the United States, which means that any data we derive from using that technology um, should be easily interpretable by practicing clinicians. If we believe, and I think we do, that the pathologic processes that encompass the beginnings of pulmonary fibrosis are the same pathologic processes that occur during the late stages of pulmonary fibrosis. And if we know that nerandomilast can slow the progression of the disease over time in people with later stages of pulmonary fibrosis, then it stands to reason that it should do it at earlier phases of disease. And so, it, whereas in the late stage of pulmonary fibrosis, you can slow down the decline, but not stop the decline. In the early stage of disease, you're basically being able to preserve normal lung function longer. And so that should translate into longer time to develop complications, longer time to need a lung transplant, longer time even to death. We're not going to be testing that in this particular study, but these are the rationales behind why we want to study it. Over a 2-year period of follow-up, I think, and just to clarify, in DROP-FPF, it's going to be a rolling enrollment, meaning that the
Hmm.
very first patient that comes in will be followed for 2 years. And then after that 2-year period of time, that person will continue to be followed until the very last person who got enrolled in the study finishes their 2 years. So it's possible we'll have people 3 or 4 years in our study. But what we hope to be able to learn from this is the speed at which these interstitial lung abnormalities worsen, the behavior of um, the patients in terms of the development of symptoms, the development of the early symptoms of interstitial lung disease that we all are taught in residency and fellowship around chronic cough and shortness of breath and fatigue. Those all have to start at some point. So hopefully, once we know we've identified somebody who's got interstitial lung abnormalities or early interstitial lung disease, we can start monitoring them at that point to see when do those symptoms pop up, because we're going to be asking them those things very specifically. It's not uncommon to get a cold and then you have a cough and that cough lasts for a few months sometimes. And most people shrug it off and say, it was a cold. But in some proportion of the population, that is the first sign of an interstitial lung disease, and we want to see if we can find it. Management right away. I think what it's going to do is it's going to provide us another piece of evidence to show that earlier identification, earlier diagnosis, and earlier intervention can improve the outcomes over time. Um, uh, where nobody is under the impression that a beneficial effect of of nerandomilast in this population in our study will automatically translate into everybody being started on nerandomilast. Like that's not likely, nor would it be indicated for the use of the drug anyway. Um, but it would provide us with some evidence. The other thing is that the study is small. It's only 80 patients, 80 subjects, I should say. They're not actually patients yet. Um, we're not expecting this to change practice. I would like to see it become involved in clinical practice. As it is now, we're using HRCTs in clinical practice routinely, and we're relying on radiologists' eyes to compare one CT to the other. But as I said before, the computer can see things much more granularly than a human eye can. So I would anticipate it should become, if we're able to show, if others are able to show the benefit of using it in clinical practice, then yeah, I think it will be adopted pretty quickly.